Hello, welcome to the next A-level lesson from the conscientious biologist, Ben Gallagher. This one's on mutations, and we look at types of mutations and their impacts on DNA. Now, I've tried to make it function as an entirely standalone lesson, but it would be really important if you've watched the proteins playlist before this. That's proteins 1, 2, and 3 that detail the process of protein synthesis. Ideally, you've watched protein biochemistry before that. But as with most topics in A-level, they really build on the foundational concepts. So if you've just stumbled across this one and you're thinking to watch it, it does assume knowledge of a lot of cell ultrastructure, DNA biochemistry and protein biochemistry. So have a watch of those if you're not sure of those topics already. Please do subscribe to my YouTube channel if you haven't done so already, and do remember I'm on Facebook and Instagram, and you can follow both of those for lots of additional bits of biological information. So let's start with the three really big questions when it comes to mutations, which are: what is a mutation, why do they occur, and why do they matter? Now, the first one's pretty straightforward to answer, because a mutation is just a change in nucleotide base sequence in the DNA. It's that simple. I know you see on lots of sci-fi films and stuff when you've got a mutation like Spider-Man mutates and you go, well, what's the mutation? Oh, I can shoot webs, or Wolverine can heal himself, or whatever. Well, actually, biologically, we're just talking about a change in DNA base sequence that could give a new protein, okay? So if you look at this piece of DNA up above me here, this is from the DNA biochemistry lesson, you can see the nucleotides, you can see them lined up in the two sugar phosphate backbones that run anti-parallel to each other. You can see the coloured complementary nitrogenous base pairs there, adenine with thymine, cytosine with guanine, held together by their hydrogen bonds. Now, what I want you to do is look kind of slightly in the middle, but slightly over towards this. Is just shift that and change it if you watch. Okay, so I'm going to switch the cytosine guanine pairing for an adenine thymine, and that's it. But it's as simple as that. That's all a mutation is. This strand above us now is now a mutant version of what was there before. So it's that simple. It's a change in DNA base nucleotide sequence, okay, or base sequence in the nucleotides. Now, why do they occur? Well, they're not deliberate. They generally occur totally randomly and naturally by mistakes in DNA replication. Now, if you think about one of the previous videos I've done on DNA replication that look like this, where you've got your replication fork, you've got DNA helicase unzipping the double strand, the DNA polymerases reading the two strands. Remember, one straight following the helicase on the leading strand, the lagging strand having to build itself in fragments. But you've still got this process. Now, those DNA polymerases have to copy thousands and thousands of DNA base pairs and get them exactly right. So for every guanine that's there, they've got to put a complementary cytosine. For every thymine, it's got to be an adenine, and vice versa. The chance of making a mistake is very high. If I gave you, a book to copy out that had 10,000 words in it, I say, can you copy that out? Chances are you'd make a couple of spelling mistakes. You might miss the odd word. You might miss out a whole sentence or something. Mistakes happen. Now, your cells have sort of safety net processes. You've got enzymes that go along the DNA checking that everything's correct, and they will correct most mistakes. But sometimes mistakes happen and they're just left for the DNA to go into the next cell. So most of the time it's entirely random. However, there are two mechanisms by which mutations can kind of be forced to happen, and it's by chemical mutagens or by radiation. Now, the word mutagen applies to anything that can cause a mutation. So radiation could be caused a mutagen. A mutagen causes a mutation. Now, there's lots of chemicals that can mutate DNA. Lots of those chemicals, you see it all over the place, can be found in cigarette smoke. So if you smoke a cigarette, you're taking some mutagens in that can mutate cells in your lungs. That mutation could cause them cancerous, which is why smoking causes cancer, okay? It's written on the cigarette packets. But there's lots and lots of chemicals in lots of things that are mutagens. Radiation, there's two main types that we talk about in biology, not quite the same words that are used in physics, but when we're talking about them it causing mutations, we talk about direct or ionizing radiation. Direct means the more powerful forms of radiation, like gamma radiation, that can directly go into the cells, hit the DNA, blast it apart, reform it, and there's errors that are left. Ionizing radiation is weaker radiation forms, like ultraviolet radiation that comes from sunlight. That radiation can hit other things in the cell, turn them into high energy ions or free radicals, and those can bang into the DNA and cause the mutations. Either way, be it by chemical mutagen, radiation, or just entirely randomly, you'll get a change in the DNA like I showed up here. Now, why does that matter? The third question. Well, it really does matter because of protein synthesis. Remember, I said it so many times in my GCSE lessons, proteins do everything. Almost every process and structure that goes on inside your cells, it's protein based. So if the DNA code is wrong, the function of the protein may be affected. Now, if you don't remember what the link is between DNA code being wrong and the function of the final protein, let's fill in those blanks. Because in the exams as well, I see this all the time. There'll be a question and the students will give me like the end point, like this in red, okay, without telling me, well, how did you get from A to D? What was B and C? Okay, so filling in the bits of information, why does it matter? Well, it's because DNA codes for the sequence of amino acids in a polypeptide. The sequence of amino acids determines how the protein folds. How it folds determines its shape, and its shape determines its function. That's why, if the DNA code is wrong, the first point, the function of the protein will be wrong. So this is a really, really important summary page of most of the information you need to know about proteins. What we're going to do next is we're really going to look at why it matters by looking how it impacts the protein synthesis. It's going to be a really quick recap. If you don't understand, if I go too fast, go back and watch the videos that relate to this. So the bit of protein synthesis that's most directly affected is the translation stage. So remember, transcription provided this mRNA copy of the template strand of DNA. That mRNA then moves out of the nucleus, through the nuclear envelope, through the nuclear pores, associates with a ribosome that will most likely be sat on a rough endoplasmic reticulum. Once it's associated with that ribosome, the ribosome will read 3 bases at a time called a codon, and it will recruit a specific tRNA molecule. You can see the tRNA up there. And that tRNA will bring a specific amino acid. Now note the differences between mRNA and tRNA. mRNA is straight, tRNA molecule still made of RNA, but it folds itself up into this clover leaf shape that the branches are held together by hydrogen bonds between complementary base pairs. So both RNA, but they form different shapes for different functions. Okay, so you recruit that tRNA, it's going to bring you the correct amino acid. We'll talk about why on the next slide. Look at what you've got there. You've got an anticodon on the tRNA that needs the complementary base pair with the codon to check it's the correct one. If it's the correct one, then the ribosome will take the amino acid off the tRNA, chuck out the tRNA molecule. That tRNA molecule can now go and find another amino acid, but of that exact same type. So if it was carrying a tyrosine amino acid, it will go and find another one of those. Okay. The ribosome then can move down the strand, down to the next codon, where it will just repeat the process. It will bring in another tRNA, it will check that that matches, take its amino acid, move down again, next tRNA, take its amino acid, move down again, tRNA, take its amino acid, until you eventually get to the end of the mRNA strand. You've got the specific amino acids that were matched up by codon and anticodon, and eventually the ribosome will come off the end of the RNA and release its polypeptide. Now, obviously, we've got a really short piece of mRNA here, only giving us a 4 amino acid long polypeptide. There'd be hundreds and hundreds of amino acids long in reality. But that polypeptide now will enter the rough endoplasmic reticulum, where it will be packaged into a vesicle and sent to the Golgi apparatus for folding and processing. If you don't know what I'm talking about the packaging into a vesicle and the Golgi apparatus, go back and watch the video on cell ultrastructure where I cover that. Now, when it goes to the Golgi apparatus and it gets processed, what we're talking about is this stuff, which again was in another video on protein biochemistry. This was showing how that polypeptide, so there's the amino acid structures at the top there in a string giving you the polypeptide. That's your primary structure, folds into a secondary structure, and then either a tertiary or quaternary structure, depending on the number of polypeptides and whether there were prosthetic groups. Again, go back and watch this video. It's protein biochemistry if this isn't ringing a bell. But it's that tertiary and quaternary structure that are so specific in shape to have really specific function. if that original polypeptide was wrong, the shape will be wrong. Now let's just drill down a bit more into that to recap how that translation occurs so we can look at how mutation impacts that. So again, this is from a previous slide on a previous video. This was from Proteins 2 about translating the code. But it needed you to understand the DNA structure, that there's a coding strand and a template strand. I'm using my simplified diagram for nucleotides here. But that to understand how DNA is translated, you need to remember that you've got this transcription process to give you the mRNA, but then you need this code, this translation code, okay? This is the universal code that tells you how those codons bring you specific amino acids. So that first codon on my tRNA over there is GCU, sorry. So you look at your translator here, you look at the G as the first position, then you've got C as the second, which puts it in this box right here, and then you look that it was a U on the end, so you know it's alanine. So we know that our first amino acid in our polypeptide is an alanine. You then just look along each codon in turn, comparing the CAG compared to this table would give you glycine, and you go along and so on, okay? Glutamine. And that's your translation to give you this polypeptide. So you get these specific amino acids from those specific codons because of how the universal code works. Now, what you need to remember looking at this, though, is there's more than one codon per amino acid. Now that's useful when it comes to mutations because it means there's a little bit of tolerance. But when we talk about the DNA code, we talk about it being universal. This code applies to every single form of life that we've ever discovered that we know about on Earth. So it's universal. Everything in our universe that we understand uses this code. It's degenerate. That word refers to there being multiple codons per amino acid. So it's not one generates one amino acid. It could be a number that generate one. So it's degenerate, okay? And it's non-overlapping. You read the first three, then you jumped to totally the next three. You didn't overlap which three you were reading. It was first three, second three, next three, and so on, okay? So really important recap there. Now let's look at how mutation messes with this. Right, we're looking at the most common mutations now, which are single base mutations. I'm going to pull up the universal code above me here, but I'm going to simplify this to try and help you to understand it because you don't yet speak genetic language. So if I said, oh, CAG, you wouldn't be able to immediately tell me what that was. So I'm going to put it in language you'll understand by using actual words. But just as codons are 3 bases long, I'm using 3-letter words. So I'm putting this little phrase up there, the fat dog ate the cat. I know it's stupid, but just think of each one of those words is representing a codon 3 bases long. These are 3-letter words, okay? So if this was my normal or original piece of DNA, what I'm going to do is cause mutations at this point where that red arrow is. So the E of the fat dog ate the cat. Now, if I show you this first one, look at what I've done there to the E. See what I've done? I switched the E for an A. So it reads, the fat dog ate the cat. Not very different. This is a substitution mutation, where I've substituted one of the bases for another one. If there's a substitution mutation, it changes one codon, one word up there. It's only changed the. And that, if I said the fat dog ate the cat, you wouldn't be able to tell if I was saying the or that. Okay, the fat dog ate the cat. Okay, it's still basically the same message, which it could be because the universal code is degenerate. Look, multiple amino acids. The and that might both give you the same meaning. 2 codons might give you the same amino acid. So a substitution mutation might make no difference at all. But of course it still could, because if it changed the codon enough, that codon could code for a different amino acid, putting a different amino acid in your polypeptide. If that amino acid plays a role in how the protein folds up, then it might fold up totally wrongly and have a totally different function. Let's look at 2 slightly more serious forms of mutation now. Try and work out what I've done here. Have a look at what's written there. See if you can work out what I've done. Remember, the mutation is where I put that red arrow at the top. You might be able to work out what I've done there in that I've deleted the E of the. Okay, so now, because there's no E, the F of fat has shifted back one, meaning every single codon is now wrong. There's been this like ripple effect. the 3 base frame with which the ribosome reads has shifted. Now this is actually called a frame shift, and you get the same thing if you add a base in. So if now I've still got the, but between the H and the E of the, I've added an N. I've now got Thenephotidogat. It's nonsense now, okay, because you caused a frame shift. The 3 base frame that the ribosome is reading has been shifted one way or the other, depending on whether it's a deletion or an addition. So this is much more significant mutation because every codon after the mutation has changed, giving a potentially very different amino acid sequence. So you've got substitution mutations, a deletion and addition mutations. But you need to know how those 3 can impact translation, and why the code being degenerate might mean that a substitution mutation might mean nothing, but deletions and additions almost always cause quite significant differences. Okay, now that new protein that could be created by any of the 3, okay, even a substitution, remember, could cause a serious thing. That new protein might not do anything, but that's really bad because your body makes proteins, or all organisms make proteins, because those proteins have a function. Well, if the new shape just eliminated that protein from having any function, it's now just a useless clump of amino acids. That's bad, because if that protein was insulin that controls your blood sugar, well, if it's a mutated form that now can't do its job, then effectively you don't produce insulin, you're diabetic. If that protein is some kind of neurotransmitter in your brain, it means your brain won't work properly. If it's growth hormone, your body's not going to grow. There's a million different proteins that could be impacted in this way. Well, not a million, but many, many, many proteins that could be impacted. If they're knocked out and you don't have a function, you're going to be missing something about how your body functions. Okay, but of course, it's not necessarily that it doesn't do anything. It might do something new. But of course, that new thing that it does, that new shape, may have a new function, but that could be good or bad. Its new function may be something really negative to the body, like it stops you metabolizing a certain type of toxin, or it stops your liver functioning in a certain way, or it stops the electrical signal traveling in the right places to your heart. It could be something awful, okay? But it might do a new function that no one else can do. That evolution, that mutation would be a bit of a jump forward in evolution. It might just be that it just does its original job in a slightly better way. It's a slightly more efficient version of an enzyme, which means you can do a metabolic process better, which is still a big advantage, which is going to cause a jump forward in evolution. Let's have a look at a couple of other less common but very, very significant forms of mutation now. So first one we're going to look at, if I put up the original strand there again, is an inversion. This is where you've got back to front bases. Obviously, it doesn't matter if you've just got one base, but if you've got a set of bases and you flip them around the other way. So for example, on here, look, I've taken the EFAT from the final E of the and the fat, and I've flipped those 4 around the other way. So I've now got Thut Afer, dog ate the cat. So those first 2 codons, because I've inverted, flipped the bases round, that's obviously had a big impact to what's going to be read, to what those 2 codons are, to what the amino acids might be. You've also got something called translocation. Translocation is where you might get a bunch of bases that for some reason jump to a different part of the chromosome or jump to a totally different chromosome. Now, if a gene or a set of bases is in the wrong place, they could knock out another gene, they could express in a different area, and something that your pancreas is supposed to do starts happening in your skin or whatever. So translocation mutations can be quite serious, and that can be any number of bases. It could be a huge, several genes, or it could just be a few bases. But you'll see here what I've done, I just took the word fat, pulled it out of being a fat dog and turned it into a fat cat. So I've now, instead of talking about a fat dog eating a cat, I've got a dog eating a fat cat. It's a very different meaning, so you can see how this could impact on the DNA. Lastly, a duplication, which could be of individual bases. So if it was AGT, it might be AGGT. You're just duplicating them, or it could be a whole section. You'll see what I've done here. I've taken the OG of dog and the A of 8, and I've just duplicated those and shoved them in the middle there. So I've now got that OGA twice in there. It's duplicated those bases. That gives me a whole extra codon. but it could give you a whole extra 20 codons or 50 codons or 1 codon. It depends how many duplicates you've made. Okay, of course, if you're duplicating 1, technically that's an addition of 1. So you've got addition mutation going on here, which is causing frame shifts. So duplications are going to cause frame shifts, as might a translocation into the position where it jumped into. An inversion isn't going to cause an additional deletion because you're just flipping them round. But you need to understand how any of these 6 mutation types that we've talked about, that's substitution, deletion, addition, inversion, translocation, and duplication, can all impact on translation, which will affect the polypeptide sequence, how the polypeptide folds, and hence the end function. Now, of course, I've mentioned in the last slide, mutation is the driving force behind evolution, which created the enormous and wonderful diversity of life on Earth. If you've never seen this absolutely brilliant tree of life diagram before, I'll enlarge it for you here. You can see right at the bottom here, the origin of life, so the first life that ever existed. Everything else on this diagram, right the way, you can see Charles Darwin as the human representative at the top there. Everything else has come along by many, many, many generations of mutations, making each subsequent form of life slightly more complex, slightly more adapted than the previous ones, giving you this vast range of life. Now, we cover a lot more on that when we do the lessons on evolution, when we cover directional selection, sympatric and allopatric speciation, and all those different things. But I just wanted you to see this brilliant diagram to see how much difference evolution has made to life on Earth. So that really does complete everything we need to do on protein. So really, you could consider the proteins lessons as 5, starting with protein biochemistry, then the 3 on protein synthesis, and now this one on mutations. Those 5 really give you a massive rounding and understanding of the purpose of DNA, the purpose of proteins, how they're made, how they can impact, and how they influence evolution. It's a massive part of your biology A-level course. Next, I want you to go to this video. This is enzymes 1, which is the mechanism of enzyme function. There should be a link for it appearing over there. As always, please do subscribe. You can click on my face up above me here to subscribe to the channel, and do have a look at my Facebook and Instagram accounts for more biology information that can back up your course and help you with any synoptic papers. Good luck. Thank you.